today we want to present to you our new products and our new portfolio, which we are launching in the occasion of the new IVD and in the new step forward for the new regulation. And I need before to remind to you that we are recording this session in order to make it on our YouTube channel, and so you can receive and forward to everyone you need, and that you can in any time do your question using the question and answer chat that you can find here in Zoom. So let's start with our webinar, and I will give the scene to Fabio for the presentation of our portfolio. Thank you.
Thank you, Valentina. Thank you, everyone, to be here for this webinar. I am Fabio Grandi. I'm the co-founder of Forbasis, and I'm glad today to introduce you to all our new portfolio kits available and the diagnostic solution available for the CA IVD market. Just a brief introduction regarding the company. Forbasis has been founded in 2013. It's a Swiss-based company. We have an Italian subsidiary where we have our laboratories and the R&D department and the production site as well. We are committed in finding advanced diagnostic solution. We are mainly specialized in the precision medicine, in particular for clinical application in different areas. In particular, the pharmacogenetics and oncology, or the genetic of the rare disease area, as well as the microbiology area, which is one of the latest we have added in our portfolio. We are an ISO 13485 certified company, and since 2013, we produce CA IVD solutions. What does it mean, advanced diagnostic solution? It means that we are producing both the kits for the library prep and the data analysis software for the data analysis. We have two main family of kits. One is the amplicon-based technology, and the other one is the new one that we are introducing now, and is the capture-based technologies. Both of these two families are made with our proprietary technology developed in our labs. And both of these two technologies has a very fast and easy workflow. That is what is usually demanded in a diagnostic lab. Most important, our kits are available and compatible with all the current NGS platforms. So we have no choice. We are not committed in particular to one particular kind of a sequencer, but our chemistry can be adapted to all of them. Besides this, we also have the data analysis software, which is required, especially for the new IVDR regulation, because we must have the overlook on all the entire process involving both the chemistry and the data analysis as well. So we have our platform that is for the secondary analysis. So it's from the FASTQ files up to the VCF files. We release a QC report reporting an annotated list of variants, including the CMG rules, and that it is available for any kind of tertiary analysis software currently available. This is a brief overview of our two families. The panel family is the family of the kits based on amplicon solution. So they are characterized being all having a very quick and easy and cost-saving protocol. It's all the protocol are less than 2 hours or 1 hour and a half of hands-on time, and they are all of this amplicon-based solution are really cost savings and are for routinary kind of analysis. The new family of kits is the capture-based kits, and of course they have a little bit longer protocol, even if we have just released the new fast version that is it's a one-day working protocol, so it's not requiring any overnight incubation. And this family of kits are including larger panels, so from 50 genes up to the clinical exome analysis. The areas of application of our kits are mainly the oncology area, where we are covering both the germline and somatic analysis. So for hereditary predisposition, we have several solutions for hereditary predisposition, as well as for somatic analysis, so products that are dedicated with an anatomopathological lab. For the somatic analysis of tumoral tissue, and in particular for the selection of the therapy. So we are involved in the pharmacogenetics area. The genetics in general, we have a solution for the routine analysis, usually based on our amplicon solution. So it's for example, BRCA1 and 2, CFTR, and other kits that I'm going to introduce you later, as well as bigger panels up to the clinical exome 1 that are mainly for clinical research, even for diagnostic application. We are also really specialized in developing really quickly custom-based solutions. So most of our kits comes from some on-demand requests from our customers, and we are open to create and generate custom panels as well. The latest areas of application we just released are for the microbiology one. We have solution both for bacterial and viral genome sequencing, for microbiome and for COVID and COVID-related analysis. Now I'm just going a little bit deeper inside, presenting you each kind of kits. Introducing first the oncological genetics kit area. We have solution, as you can see, different kind of kits, the panel kits that are based on and the screen as well in amplicon solution, the pro kits that are capture based. So we have solution for germline analysis and predisposition, for routine analysis and clinical research, and we have different solution specifically dedicated for somatic analysis. Some of these kits, like HIVA Pro, for example, or RASPO Pro, can be applied both for the two solutions as well. The BRCA kit is actually one of the older kits we have in portfolio since 2015. Is a IVD solution. We have two different solutions. This one is particularly dedicated for genetic analysis, so for routine screening. Since it's a very quick and fast protocol, and it's amplicon based, and this is for the identification of BRCA1 and 2, SNPs, indels, and CMVs as well. The target customers are, of course, the public and private diagnostic genetic labs. This is BRCA panel. This is one of the most common and known tests available in NGS since the last 5 years. But now the demanding of the genetic lab are more orientated to some bigger solution. So some solution that is including not just the BRCA1 and 2 genes, but also all the BRCA1 and 2 related genes. That is a family of about 50 genes that are all of these are present in the HIVA Pro solution. It's a capture-based solution, so the protocol can be done in one day, one single day. And it can be used both for hereditary, so germline analysis, and somatic variance profiling. In particularly 99% at least of the known and common cancer-related disease known today. So these kits can be applied both for a genetic lab as well as in an anatomopathological lab, because it's a very versatile kit, and this it's one of the reference kit for the routine analysis of the BRCA related and homologous. You see the homologous recombination repair kit genes related. This solution is very for a niche, of course, but this comes from a specific request from some of our customers, and this is very targeted solution for the identification of all the mutation in all the genes that are related with the neurofibromatosis and the rasopathies in general. So it's including 29 genes. As you can see, is in the list on the table on the right of the screen. It's a capture-based protocol, so it means that it's covering all the codifying region of the genes, including the padding region around each exons. And is for routine screening of all the variants related with neurofibromatosis and rasopathies in general. Okay, now we are leaving the genetic aspect, so the hereditary aspect that can be used also for somatic, and we are jumping into something that is really committed with the somatic and so for the solid tumors and the anatomopathology lab. The first kit is the BRCA screen that is very similar to the BRCA panel, but is really more focused for a somatic analysis because it's based on a different technology that has been optimized to work even with the very damaged FFPE samples. It has been validated for the analysis of breast and in particular for the ovarian cancer tissues. And this including BRCA1, 2, as well as TP53. And this is a very targeted kit for the precision medicine application of the cancer therapy selection. So this is why we are presenting besides the BRCA panel, because the BRCA panel is for routine analysis. This is for the anatomopathology lab, even if it can be used for germline analysis, because it's validated as well for germline analysis. We believe that it's more focused for the anatomopathology lab. The Bankit panel is one of another historical kits from Basis that is in our portfolio since 2017. I mean, it is including the most common hotspots required for the therapy selection and in colon cancer, metastatic cancer, and in non-small lung cancers as well. And it's including the main common hotspot of KRAS and RAS, BRAF, EGFR, and PIK3KA gene. And this is for a routine analysis in an anatomopathology lab. And this is a very cost-effective kit, even if it's more targeted for the medium size of anatomopathology, because sometimes the very small anatomopathology they don't have enough number to be to have an NGS system. But with the new benchtop sequencer coming from the main company, this kind of kits will be even much more, desirable even for the anatomopathology lab, which have access to NGS technologies. The big brother of the bank kit is the PanCancers Pro kit, which is something that is now demanded from the big anatomopathology lab, which already are used to work with the NGS sequencers. And it's including 95 genes, the same genes that were present in the bank kit, but extended, so including all the codifying region and the pending region. 95 genes that are related with are shown to be relevant with solid tumors analysis and for the cancer therapy selection. So it's an extension of the bank kit based on capture-based technology. As well as the bank kit, it can be used for germline and somatic analysis, And it's also validated to analyze the CMVs from this tissue, even if it's a somatic analysis. This is the list of the kits, but we will you will find it in the very next days on our website when we are going to officially release these new families. So this is just an anticipation, and it will be released in the very next days. You will find all the documentation on the website. The last kit related with solid tumors, is the HRD kit that is very similar to the HIVA Pro, so it can be used instead of HIVA Pro, and is more focused in finding all that alterations. So it's single nucleotide polymorphism, even in structural variation, that are related with the HRD score prediction and evaluation. So it comes together with our kit, with our software Forever that I'm going to introduce you later, and it will allow to evaluate the HRD score. So it's a kit. It's a kind of kit that is going to be more and more demanded from the anatomy pathology, and it's already available. It's a capture-based protocol, but it takes, I repeat, just one working day. The last kit regarding the oncology area is the HematoPro. That is not for solid tumors, of course, but is for the analysis of that complex genomic variants that are associated with the lymphoid, myeloid disease, and with the MPN in general. It includes 137 genes, and of course, it's a capture-based protocol, and it's for germline and somatic analysis of all the SNPs, the indels, the CNVs, and related with that complex genomic variants. And the starting samples can be, of course, blood or body tissues of any kind, because all our capture-based kits can work even on somatic or FFP samples. That's the list of the gene included in the HematoPro kit. So the other big area of application of our kits is the genetic and the rare disease. We have to split, we decided to split the list of our kits, even if they have the same technologies and amplicon-based and capture-based, by areas of application. So the genetics and rare disease includes, as well as for the oncology one, a list of kits that is dedicated for routinary analysis. So it's amplicon-based solution, and all these kits share the same fast, easy, and cost savings protocol. And we have a family of kits that is capture-based and comes from the Clinex Pro, that is the our clinical exome kit that is including, we will show you all the homing genes and the GWAS gene, about 26,000 genes in general, and all the other families, that is cardio, ped, nephro, retina, and well, that is a sub-part of the Clinex, the original one. And they are smaller kits that are dedicated for specific clinical applications. So the CFTR is like likewise the BRCA kit is the other most common routinary analysis that is based on NGS technology since 2016, 2015, even earlier. And our kit is an amplicon-based kit, and it comes with the Forever solution that allows the analysis of all the variants, intronic and exonic variants related with the CFTR gene that are related with the cystic fibrosis. And we will apply the demanded filters regarding the first and the second level of analysis. It's applicable with the Forever solution. So it's a very fulfilling kit that is has all the characteristics to be applied in a routine screening for the CFTR or genetic or cystic fibrosis genetic analysis. Usually the screening is about prenatal or more commonly for newborn. Each newborn has been screened, and these kits can be used also starting from the kind of samples that is usually used for the newborns. So the small tissues, and they can be used in this in a IVD solution already validated, and this is in our portfolio since the last 4 years at least. The thalassemia panel as well. The next one is more focused keys for something is a niche, but it's highly demanded for the routine analysis. This kit is an amplicon-based kit, so it's very fast, as exactly the same protocol all the other panel kits, and it's including the HBB, HBA1, 2, HBD, HBG1, etc. All the genes that are related with the analysis of thalassemia. Both alpha and beta thalassemia. It allows the screening of the single nucleotide polymorphism as well as the copy number variation that is really important regarding the analysis of the thalassemia. The other kit is always based on the amplicon solution technology. So it's the FH, we call it, is for the characterization of the familial hypercholesterolemia, and it includes 9 genes that are usually identified for the, I usually investigated for the identification of some polygenic SNPs, and also for some SNPs related with the prediction of the statin response to the, with the proper targeted therapy choice and adaptation. This kit is actually the last one. It is a very interesting kit because it's already been part of some initially multicentric clinical trials in Italy, but it's a very important kit because it's a kit that is including, we have developed it together with the University in Rome of Tor Vergata and Bambino Gesù Hospital. And it's a kit that is including about 80 hotspots that are related with the predisposition, so for the resistance or susceptibility to respiratory infectious disease in general, and in particular related with the COVID-19, of course. These kits come with the forever solution that is under patent now solution that is able to release a prediction score regarding the, so a risk score regarding the possibility to be resistant or to be highly susceptibility to the respiratory infectious disease. We know this because it's these variants are common, and it's part of the you know, the scientific literature till now, till 2022, and some of this hotspot was discovered in the latest period from University of Tor Vergata and Bambino Gesù as well. We included it, and we have an algorithm for the evaluation of the different combination of the variants, and the risk score is more than 95% accurate as well right now. So the risk panel is the last one with an amplicon-based solution. I repeat, all these panel kits are sharing all the same operative protocol. It is less than 1.5 hours hands-on time, and it's something like 6 hours of total turnaround time from the DNA to the library ready to be sequenced, and can be are compatible with all the current platforms. So for Illumina as well as for Thermo Fisher, etc. All these kits, so the IVF Pro down to the clinics kits, are based on capture-based solution. The capture-based solution, repeat, is a fast solution we developed, and it doesn't require an overnight incubation of the probes, but it's just 1 or 2 hours. So the whole protocol can be done starting in the morning before right to the evening, in a working day or a 2.5 working day. The IVF Pro in particular is dedicated, is a carrier screen kit. So it can be applied for the carrier screen analysis of a single person, or can be related with the in vitro fertilization. So for the screening of the 2 donors, looking for autosomal recessive inherited disease, or rare conditions, or X-linked diseases, or up to cancer-related diseases. It's including 61 genes, several hotspots, and that is covering more than 300 different rare diseases. The target customer, of course, is the genetic laboratories, but also the IVF specialized centers, because it is a very oriented kit for this kind of application. And here is the list of the gene included in the IVF Pro. Another kit that is really oriented with a particular clinical application is the CardioPro kit. CardioPro kit is one of our latest release, and is 100% following the latest European guidelines regarding the cardiomyopathies, genetic related analysis. So it's including more than 200 genes, including all the genes that are suggested and required from the latest guidelines. And so is, I think, right now the only one that is 100% following the guidelines, because all the other kits that must be updated from the competitors, I think. Is capture-based kits. All our capture-based kits share all the same protocol, and as well as for the Amplicon one, they can be mixed in the same analysis. Customer can prepare different libraries from different applications. We will provide a table for the balancing of the different libraries, depending on the number of samples and the kind of application. And all these libraries can be mixed in a balanced way and uploaded in the same cartridge. So it's a cost-saving solution. And this is just because all these kits share the same technology, and that what allows us to mix them and to provide for all these solutions the same, even the operative protocol. And that's a list of the genes included in the CardioPro solution. A bigger one kit, okay, that is more than 450 genes, is the PID Pro, the PID Pro, that is for the primary immunodeficiency disorders genetic related analysis. So it's oriented for the neonatal screening, because the neonatal screening actually represents one of the most effective tools now we have for early diagnosis. And it's key that once again is 100% following the latest guidelines for this kind of application. The list of genes starts to be even too much to be included in a table, so it's this is the last one, because the NEFRO one, of course, and the RETI one, they are a very small panel, but the Kleenex one is, we cannot include all the genes in the table, of course. The NEFRO one, okay, is an on-demand kit. This is very peculiar example, together with the Retina Pro, that is the following one, of a collaboration with some reference centers, some in some situation can build to a very specific and personalized kits and customized kit, and allowing this reference center to have a specific solution. So they don't have to use a bigger solution if they don't need to. So they can save costs about sequencing, about data analysis, and having a very more fluent workflow for all the diagnostic they need. The NIFRO is including 92 genes, and it's generally related with the polycystic kidney in general and nephrotic condition in general, of course. So for the nephropathies. The other one is the Retina Pro, that is 107 genes, so more than 100, but it's more or less of the same size of the NIFRO Pro. And this for retinopathies and for all the overlapping condition related. So this is an example of we can create kits for specific clinical requirements. So just ask us and we can create some custom kits that will come with the secondary analysis solution. This is one of the most important aspects. So the last one of this big family is the Clinics Pro, and this is something that we have developed in the last 1 year, and now we are going to validate it, and there will be a IVD solution before the next May 26. Because the clinical validation is undergoing, and it's going to be fulfilled before the end, before the next 1 week or 2 weeks at least. It's apparently it's a very big size kit to be a clinical exome because it's included 26,000 genes, but it's not like that because it's a very balanced solution, and the uniformity of these kits allows us to run more or less the same number of samples that can be run with the 5,000 or 7,000 solution kits. So we can run, for example, a trio, so 3 samples in a V3 on a MySec. So it's very uniform and very effective, cost-effective solution, but it's including more than 10,000 genes from OMIM. It's including all the alternative transcripts and pseudogenes, and it's including all the GWAS genes. This means that this solution will be the reference one for the clinical exome for the next years, because it's already including all the genes, the GWAS one, for example, that will be included in the OMIM. So will be an always updated OMIM kit, and they will be always including all the alternative transcripts and pseudogenes that is required for a very detailed analysis. It's a capture-based kit, and this has been validated with a very fast protocol. Only 1 hour of incubation for the probes is required for having a solution, having a very good results and uniformity. So 1 working day from the morning to the evening, and you have the library ready to be sequenced for a clinical exome, and with a very cost-effective sequencing solution. The last area of application, so it's just 2 kits because it's a new area for us, and then I will go introduce you the last topics, that is the forever solution. So the last area of application is the microbiology kits. We have 2 solutions in particular. One is for microbiome analysis, and one is for COVID panel that is required in the last period for the analysis, the sequencing of the viral genome. The microbiome panel is a CIVD solution that is actually including all the hypervariable region of the 16S DNA gene from the bacterial DNA. So it's including from V1 up to V9 genes, region, sorry. And so it's able to analyze different kind of microbiome, of human microbiome. So we have a region specifically, for example, the V1 and V2 that are more specific for the vaginal microbiome, the V3 and V4 for the intestinal one, the V6 and V7 for the buccal and the oral one, and for the lactose enzymes. And we have also validated it for the surfaces and soil microorganisms. So in order to understand the nosocomial infection, our hospital surfaces, for example, and or soil microorganisms. So it's a very versatile kit. And in the same kit, it comes also with a separate oligo pool, including ITS1 and 2. That is for fungi and moldus application that is not yet being validated. So the IVD solution is from V1 up to V9, but in order to have a very extensive solution, including in the same kit, we included also these two other regions, ITS1 and 2. So it's it's a file for the analysis and phylogenetic characterization of different kind of human microbiome communities, and it's the target customer are very different. It can be microbiology labs usually, but it can also be some private labs for other kind of application that is more related with wellness and not just with the diagnostic, even if it's an IVD kit and it comes from the diagnostic area, of course. We developed a specific bioinformatics solution for the microbiome panel. The data analysis is based on Kimi2 software, which is a gold standard, of course, and the classification is based on the Silva database. And of course, it's always updated with the latest version. Now it's 2022. It's updated every about 2 or 3 months. And it's always to upload the files in batches. And so it would be, it's a very easy and very usable solution for the identification and characterization of the different phylum families and species and so on of a microbiome community. The last kit of we are going to present today is the COVID panel. It's an amplicon-based solution, so it's a very quick and fast operative protocol. It allows to sequence the entire SARS-CoV-2 viral genome, and it's updated with the latest version of Primus that allows to detect all the known variants, including the Omicron or the latest one that has been released. It is a CA IVD solution. It is already always updated with new variants when new variants will arise in the community. So it will be updated if it's required in some primers. And the hands-on time is really, really short because we know that usually the sequencing of COVID is in a large batch of samples. So all our kits can be easily implemented in automatic systems for automatization of the protocols. We already have this kind of collaboration, but nothing official. So they work perfectly. We just have to update the script for each kind of different instruments for automatization, or even if they are used with a manual protocol. In particular, the COVID one is really hands-on time saving time, of course, because in less than 1 hour, and it's a very fast and quick protocol we developed specifically for this application. So the last topics before I close with this presentation is the Forever software. The Forever software is our bioinformatics platform, and it's the bioinformatics platform we developed for the validation, internal validation, and for the analysis, the secondary analysis, all our kits. This is just because the IVD and VDR regulation required to have some fixed solution, both for the chemistry and the secondary analysis as well. So we developed the Forever, and it is a really user-friendly interface where you can upload the FASTQ files coming from any kind of sequencer, so from Illumina as well as from Thermo Fisher or other kind of sequencer, and all the pipelines from our kits has been validated with the software that is so fulfilling a CA IVD solution, and the output is a VCF file or an annotated file. So the TSV file that is including also the HEMG annotation and HEMG classification. So it's the output file, so the BAM and the BY file can be downloaded as well, can be uploaded in any kind of a tertiary solution, software solution. The fact that we use this kind of application is increased a lot the accuracy of the results, because we know all our kits internally, we know the different kind of analysis that is required to be more efficiency to our kits, and so this allows us to offer a complete solution with a minor cost, because the cost of analysis is included in the cost of the kits. It's based on a proprietary software, so it's a proprietary software forever. I repeat, it's independent from the sequencing platform we used, so it can be used from FASTQ files, because our kits can work on every platform, and the software can work as well. It gives output about the coverage, so a QC, BAM, BY, DCF, and TSV file as well, that can be uploaded to any tertiary solution. Okay, this is what the Forever software is already available, but we are always in a development. This is just an anticipation that is will be in the next release. So a more rapid visualization of the VCI file annotated in the browser. We are going to release API in order for a direct transfer from base space. So from your base space account, it can be taken the FASTQ files directly. You have not to move it to upload and download it. We will release some videos regarding in support, so for marketing and for user manual instruction. We are going to release also and to conclude the validation of the CMD's calculation. And this is just for distributors, of course. It's a backend we can manage with the direct token claim. All our kits are, of course, validated also in a tertiary solution. In particular, we have a long time collaboration with Varsum, the Varsum Clinical, where we have validated both the secondary and the tertiary analysis. So you can work with Forever and then work with the Varsum for the VCF, or you can upload directly the FASTQ files. And it's validated both for somatic and for germline application. And the same way, we also tried and adapted our VCF output and tested. Of course, we know that several customers are using it with other kind of software. So just to report it, we are already working with Genix. That is very interesting platform for the annotation. In particular, is dedicated for exomes and genomes, clinical exomes. And the input is the VCF, and they have the ACMG classification from germline. We worked also with the Genome, even also for always for the tertiary analysis. So for exomes, genomes, and they work also with smaller gene panels, and they can both work for somatic and germline. The last one that we can suggest is Franklin by Genux. That is, it should be at least with the basic solution as a online free available tools. where you can upload the VCF and it can work both for somatic and germline analysis. So just to show you that the integrated solution we are offering, the kits together with the secondary analysis, is a complete solution and it can be easily adapted to several different kind of other commercial software. We already talked about Varsom because we are working with Varsom. We validated already with our pipeline with Varsom, but we work also with several software. So it's this is not related, the BCF file analysis annotation with the CIVD certification. So the kit and the solution is CIVD. You can apply the VCF on any kind of tertiary analysis software you want to use or you prefer. So that was the last slide. Thanks for watching, for listening, and I'm available for any kind of requests and answers.
Thank you, Fabio, for this presentation. I know that the presentation was quite long, but we wanted to give you first overview of our new portfolio. Distributors from years of basis probably presence of some panels like Eva Screen that become Eva Pro, and that we have both BRCA screen and BRCA panel, and we probably in the next year convert BRCA screen in a panel solution. Why panel solution? Because the panel solution comparing with the screen solution is faster, and with a simpler protocol, which gives you the possibility to in a day arrive to the sequencing. And yes, we have a lot of questions. Much of them are about HRD score. Okay, we are working now on HRD score with a clinical center in Italy. We are validating it, and HRD score works with HRD Pro and Eva Pro panels in particular, and is available for breast, ovary, and endometrial cancer now, but we are working also with other type of cancers, so other type of scores. And how will HRD scoring be done? Will it be on all genes? No, and yes. HRD score will be calculated on the HRR genes of the panel, and we will use a group of SNPs that we specifically calculated for the calculation of the HRD score. So the panel is the same for the customer, but putting the FASTQ file in our software, the calculation will be available. How much samples are needed for CMV in one run? Yes, normally 5, almost 5 samples in one run. Of course, if we are speaking about same panels, same technician, same machine, we can you can suggest to your customers to mix together different samples from different run, but this is something we have to see, and what I can suggest is every time no less than 5 samples. Which panel can be recommended for genomic instability? Well, this is a great question and very difficult. Recommended for genomic instability, probably it means some more explanation of what you need. We can, of course, speak in a different chat or in a different, you know, call about this. In this first answer, I can say Kleenex Pro, because it's enough extended for you to see every structural variant in the genome. But of course, it depends on the application. You can do something Fabio said, but I want to remind you is that every protocol of our Pro family is fast and is tested and validated both on somatic and germline samples. So it can be used also for FFP. Oh, Fabio, we have another question. This is for you. Any update on fusion panels? Fabio is laughing because we had a quite extensive discussion about fusion panels. Yes, we are working on it. We are working on it with the capture-based. So it will be a capture-based panel. The preparation of the Fusion Pro, probably we will present you in the summer the Fusion Pro, and it will be from RNA. And the preparation will be separated from other kits, but you will in the end can have the possibility to suggest to your customer to run in the same run with other kits, something like pan cancer and so on, in order to give a complete solution also with fusions. I'm going rapidly with your answers, but if you want, of course, to go deep in something, please go on in writing, in asking, or send us an email and we will organize a more deepened explanation. How about mitochondrial genome panel? Yes. Also with fusion, we are working also on a MitoPro panel. We are testing and we are testing it alone. Is a Pro panel, so capture based. We are testing it alone, or there is the possibility to use it with another kit, for example, Kleenex Pro or Cardio Pro, in order to see mitochondrial disease with cardiomyopathies. And in this way, you can have a complete solution also with mitochondrial genome. The MitoPro protocol is a fast protocol also, but if you are focused on the mitochondrial genomes, probably, yes, not probably, but the hybridization time will be quite longer, will be 4 hours, because the target is quite smaller. Can we run our panel on MGI systems? Great questions. Yes, you can. We have done some tests, and yes, you can. And you can with Ion Torrent, you can with Illumina, you can with MGI, and I will see to you, but it's something very new. We will give you the final info. You can analyze with our Forever. If the number of clinical relevant genes is approximately 5,000, then how it is possible that in the Kleenex panel there is 26,000 genes present? Yes. Clinically relevant genes is approximately 5,000, yes, but OMIM recognized genes are quite 10,000, and we decided to put in this extended clinical exome also CCDs and different transcripts that are relevant or were published or were described in GWAS studies in order to have a very complete solution also in the next years, because this panel is a CAVD panel, so it will be closed, and so with this configuration for, I hope, the next 5 years. Are you thinking to publish TMB panel? What do you mean about TMB panel? If you speak about the TMB calculation, we are working on it and validating it also for forever. In particular with our HRD and EVA Pro, but also with pan-cancer, of course. When will be all new products ready for sale? Oh, well, it depends on when you will send to us the first order. Of course, we are ready.
Yes, we are just to clarify that, okay, all these kits, a large part of these kits will be is under clinical evaluation and will be validated before May 26. So if you want them as a fluoro solution, it's immediately available. If you want as an IVD solution, you have to wait for June, the 1st of June, because okay, 26th of May, okay, after 26th of May. But the official launch of the IVD solution will be on the 1st of June. But the research is only kits, that is exactly the same solution, is already available, of course.
Any indication by when the HRD Pro scoring software will be ready by? Yes, we will be in a beta test in June, and it will be available from September. I hope I answered to all your questions.
I want to thank especially the anonymous one which wrote, "Forever is great news." I'm agree with you, my friend. Forever is a very great news because it's completing our offer and allows us to work with our partners with no particular boundaries, but with the most effective solution based on VICF WAV files, and so it will increase our collaborations and our the strength of our solution a lot, the Forever solution. So thank you. I agree with you.
Okay. If there are no more questions, of course, you can anytime write to us an email and we will be glad to reply to you. And probably, not probably, but for sure, we will organize monthly webinars to go deepen in 1 or 2 products for every family, in order to explain to you more in details the technical part, the data analysis, what we are expecting, and every news about Forever and so on. Okay, so thank you for attending this meeting and for all your questions, and see you with our next 4 Bases meeting.
Thank you very much. Thank you. See you next time.